Welcome to Developmental Biology. Today, we are going to explore one of the most fascinating structures in nature, the egg membranes. Think of the unfertilized egg as a highly valuable treasure carrying the genetic blueprint for a new life. To protect this treasure in a dangerous and unpredictable world, nature has evolved a brilliant system of shields. We will uncover what these membranes are, how they function, and even look at the mathematics of how they keep the developing embryo alive. Let us dive in. Now that we know what we are studying, let us build an intuitive picture. Imagine a king's crown sitting in the middle of a dangerous world. You would not leave it exposed. You would build walls, moats, and gates. In biology, the ovum is that precious treasure. The egg membranes act exactly like those castle walls. They provide multiple layers of defense against physical damage and drying out. More importantly, they act as a highly secure gate, ensuring that only one specific, highly qualified messenger, a sperm from the exact same species, can enter. Without these shields, the fragile egg would be easily destroyed or fertilized by multiple sperm, which is fatal. Building on our castle analogy, let us look at the formal biological definition. In developmental biology, egg membranes are defined as the specialized extracellular layers that surround the plasma membrane of an oocyte or ovum. Scientists systematically classify these envelopes into three distinct categories: primary, secondary, and tertiary membranes. This classification is not based on their thickness or chemical makeup, but rather on their biological origin, specifically where and by what cells they are produced during the mother's reproductive process. Each layer is biochemically distinct and serves a highly specific function at different stages of development. Let us break down these three categories to understand exactly where they come from. Primary membranes are secreted directly by the egg itself while it develops in the ovary. Examples include the vitelline envelope in amphibians and the zona pellucida in mammals. Secondary membranes, on the other hand, are secreted by the ovarian follicle cells that surround the egg, like the tough chorion found in insect eggs. Finally, tertiary membranes are added much later by the oviduct or maternal accessory glands after the egg has left the ovary. This includes the thick jelly coat of frog eggs and the hard calcareous shell of bird eggs. Having discussed the three layers, let us visualize them together in this cross-section diagram. Right in the center, you can see the nucleus and the cytoplasm, which make up the core of the oocyte. Immediately surrounding the oocyte is the primary membrane, labeled here as the vitelline envelope. Just outside of that, we see a layer of cells representing the secondary membrane, formed by the follicle cells. Finally, the outermost, thickest layer is the tertiary membrane, shown here as the jelly coat. This diagram beautifully illustrates how the egg is nested safely within these concentric, protective shields, ready to face the external environment. Now that we can visualize these layers, let us understand why they are absolutely critical for survival. First, they provide a species-specific recognition system. They act like a biological lock that only a sperm with the correct molecular key can open, preventing cross-species fertilization. This is especially vital for animals like fish and frogs that release eggs into shared waters. Furthermore, once a single sperm successfully enters, these membranes rapidly undergo a massive structural change. This change instantly blocks any additional sperm from entering. This life-saving process is known as the block to polyspermy, ensuring the embryo has the correct amount of genetic material. Beyond nature, these biological locks have profound implications in modern medicine. In humans, the primary membrane is called the zona pellucida, and understanding it is fundamental to treating infertility. During in vitro fertilization, doctors sometimes encounter situations where a patient's sperm lacks the natural enzymes required to penetrate this tough membrane. To solve this, medical professionals use a remarkable technique called intracytoplasmic sperm injection, or ICSI. Using a microscopic needle, they physically bypass the zona pellucida to deliver a single sperm directly into the egg. This shows how mastering developmental biology directly translates into life-changing medical technologies. While we have focused heavily on protection, this brings us to a critical biological challenge. The egg membranes cannot be completely sealed off from the outside world. The developing embryo inside is a living, breathing entity. It requires a constant, steady supply of oxygen to survive, and it must continuously get rid of toxic carbon dioxide. Therefore, these protective walls must remain permeable to essential gases. To understand exactly how the physical properties of the egg membrane dictate the survival of the embryo, we can model the movement of these vital gases using a mathematical principle known as Fick's first law of diffusion. This mathematical principle we just mentioned gives us a clear window into the embryo's breathing process. The formula states that J equals P times A times the difference between C out and C in. Let us break down every piece. J is the net rate of diffusion, telling us how much gas crosses the membrane per second. P is the permeability coefficient, which measures how easily a gas passes through the specific membrane material. A is the total surface area of the egg. Finally, C out and C in are the gas concentrations outside and inside the egg. If the membrane hardens, P decreases, meaning the diffusion rate J will also drop, potentially suffocating the embryo. Before we move on, let us see this formula in action with a concrete biological example. Imagine a frog egg, newly laid in a freshwater pond. We are given a permeability coefficient of 0.05, a surface area of 0.12, an outside oxygen concentration of 0.30, and an inside concentration of 0.10. We want to find the initial rate of oxygen diffusion. First, we substitute our given values into Fick's law. We calculate the concentration gradient by subtracting the inside oxygen from the outside oxygen, giving us 0.20. Multiplying the permeability, area, and concentration gradient together, we get a final rate of 0.0012 micromoles per second. Physically, this means a steady, life-sustaining flow of oxygen is reaching the rapidly dividing cells. Now that we have calculated the normal breathing rate of an egg, let us look at what happens the moment fertilization occurs. Immediately after a sperm successfully enters a sea urchin egg, the egg undergoes a massive structural change called the cortical reaction. During this event, the primary membrane lifts away and hardens into a tough fertilization envelope. As we discussed earlier, this is the block to polyspermy, designed to keep other sperm out. However, this hardening process naturally decreases the membrane's permeability to gases. The egg is making a biological trade-off. It gains immense physical protection, but it sacrifices some efficiency in gas exchange. This biological trade-off we just discussed leads us to our second calculation. Suppose the cortical reaction causes the permeability coefficient to drop to 0.01, while the surface area and concentration gradient remain exactly the same as our previous frog egg example. What is the new rate of oxygen diffusion? We use the exact same diffusion formula, but we update the permeability value to reflect the hardened fertilization envelope. We plug in 0.01 for P, 0.12 for A, and 0.20 for the concentration difference. Multiplying these together, we find the new diffusion rate is 0.00024 micromoles per second. The rate has dropped significantly, showing that the membrane must strike a delicate balance between being tough enough to protect, yet permeable enough to breathe. We have covered a lot of ground today, so let us summarize the most important ideas. First, egg membranes are not just static walls. They are dynamic, multi-layered shields essential for the survival of the embryo. We learned they are classified into primary, secondary, and tertiary layers based on where they are produced in the mother's body. Second, these membranes actively regulate sperm entry to prevent cross-species breeding and lethal polyspermy. Finally, through Fick's law of diffusion, we saw the delicate mathematical balance the egg must maintain, hardening to protect itself while remaining just permeable enough to manage the diffusion of life-sustaining gases. You now have a solid foundation in the protective shields of the ovum.